The purpose of this study was to test the idea that infections interfere with the prenatal formation of the nervous system, and that this interference causes devastating birth defects. Neurodevelopmental disorders are lifelong disabilities in learning and behavior because the brain has not developed normally. The picture shows microcephaly, a brain that is too small. There is no effective treatment. Some disorders only occur in one individual, while others occur much more often. In either case, it is extraordinarily difficult to understand what causes the disorder. The data used in this analysis comes primarily from these 3 published studies, all of which were very, very excellent. Some neurodevelopmental disorders are caused by deletion of a large chromosome segment, for example, a deletion on chromosome 18p, the short arm of chromosome 18. Many functions have to be coordinated as the brain developed, so I wondered if the loss of a piece of a chromosome causes the loss of a set of coordinated functions. The functions I tested for are shown here. It turned out that the loss of a chunk of a chromosome like chromosome 18p did cause the loss of a variety of coordinated functions. Many of the genes on this lost chromosome piece were responsible for more than one function. In the picture, genes needed for the nervous system are color-coded in blue. Genes for the immune system are pink, genes involved in circulation are yellow, and structural genes are gray. In all, consistent results were found for 6 different chromosome losses. Each of these 6 losses causes the loss of neurologic functions. The conclusion from these sets of experiments is that genes required for converging functions in neurodevelopment are arranged near each other in close clusters. The loss of these gene clusters explains why the errors in neurodevelopment are so devastating. Next, the work turned to what causes these kinds of disorders. The central idea was that infections are involved. Infections are known to cause problems in how the baby develops. It has been known for a long time that an infection like human herpes virus 6 can cause chromosome breaks and then probably loss of pieces of chromosomes. One basis for the interference of infection with human DNA is that foreign DNAs match segments of human DNA that are repeated millions of times. The top panel shows the position of the starting point of significant matches between human DNA and infection DNA. This occurs in a large segment of chromosome 4, for example. Deletion of a large piece of chromosome 4, including this segment, causes a problem in neurologic development. In the bottom of the graph, the position of repeated DNA elements are shown. And these repeated DNA elements line up well with the position of infection matches. If there is an infection or some reservoir of foreign DNA that matches to human repeated DNA, then this would favor interference with human neurologic development. When a large chromosome segment is deleted, new junctions are generated, and similarities to foreign DNAs can then become very different. For example, when the chromosome segment 4q34 is deleted, a new junction is created between 4q33 and 4q35. When this piece of chromosome 4 is deleted, normal matches to microbial DNA undergo massive changes. The graph shows that many normal matches to microorganisms are lost, and what is left becomes very, very different. Abnormal DNA sequences in areas of DNA that are involved in shifts of pieces of chromosomes have been identified as drivers of neurologic disease. Many abnormal DNA sequences in some patients are similar to infection. I want to show data for patient 154 as published by Reedon and coworkers in 2017. This patient had speech, motor, and cognitive delays and unusual structural features. Here is about an 800 base pair chromosome abnormality in this patient. This abnormality is near one of the breaks that occurred in the baby's DNA. Two parts of the sequence match pieces of human chromosome X. An insert between them does not match human sequences. The first two of these abnormal matches are parasitic roundworms, and the last two represent yeasts. The odds that this match is due to chance are infinitesimal, being less than 10 to the minus 10th power. Errors in neurologic development up to this point occur in one individual, and they are very rare. Next, the study turned to errors that can be found in more than one person. One of these recurrent disorders is characterized by a particular exchange between pieces of chromosome 8 and chromosome 22. This chromosome change predisposes to other change. Here you see that an affected child has very different similarities to foreign DNA than the mother. So the study findings applied to recurrent and familial neurologic disorders as well as private or individual neurologic disorders. In summary, the results of this study suggest that infection control and genetic testing can prevent some devastating birth defects that otherwise have no effective treatment. Thank you for listening.